Today, I would like to present my research in the title, The Effectiveness of Asiaticoside on Osteogenic Gene Expression and Mineralization in Aging Human Periodontal Ligament Cells. And this is the list of the contents I will present today. Periodontal ligament is the periodontium component that helps support the tooth. It also provides nutrition to the periodontium and has regenerative ability. Human periodontal ligament cells can produce inflammatory cytokines and build ligaments and fibers. Stem cell-like traits of periodontal ligament cells can differentiate into osteoblasts and produce alveolar bone and cementum. However, the aging condition of one individual leads to the alterations in periodontal ligament cells. The aging periodontal ligament cells have a low proliferative rate compared to young human periodontal ligament cells. Aging cells have high levels of inflammatory cytokines such as interleukin-1 beta, interleukin-4, interleukin-6, prostaglandin E2, and cyclooxygenase 2. Regarding the regenerative ability of the cells, aging cells express decreased level of osteoblastic genes, such as collagen 1, alkaline phosphatase, osteocalcin, osteopontin, dental matrix protein 1, and bone morphogenic protein 2. The lower rate of regeneration combined with long-term accumulation of dental plaque and calculus may lead to a higher risk of tooth loss in elderly individuals. Loss of teeth not only affects medication ability, but for the aging population, the number of teeth remaining also enhances the risk of frailty in elderly individuals. It would be better to find a procedure or material that would prevent the process of tooth loss or promote periodontal regeneration. Asiaticoside is a pentacyclic 30-carbon skeleton with a glycosylated shape that is extracted from Centella asiatica, known as baobab in Thai. It has potential in promoting wound healing. Asiaticoside was reported to have the ability to promote periodontal regeneration. Asiaticoside can promote alkaline phosphatase activity and increase osteogenic gene expression, resulting in promote of bone mineralization. However, the effect of asiaticoside on aging human periodontal ligament cells is unknown. This study will be the first to determine the effect of asiaticoside on osteogenesis-related genes and mineralization nodule capability in aging human periodontal ligament cells. From April 2020 to December 2020, teeth with no pathologic symptom and was confirmed periodontium condition using radiographic image were recruited from patients at 60 and above, according to the dental treatment plan. This is the process of all methods we perform. The cells were explanted from aging periodontal tissue and culture until they reach passage 4 to 7. The viability test of the aging cells treated with 12.5, 25, and 50 micromolar asiaticoside was conducted using the NTT assay at 72 hours. With the viability test result, we select the proper concentration of asiaticoside to perform the following experiments. The aging cells were maintained in osteogenic medium, and the osteogenic genes expression level was evaluated using real-time PCR on days 1 and 7. Mineralized nodule formation was analyzed using alisalicate staining after the aging cells were incubated in asiaticoside for 14 days. The viability test results show that Asiaticoside did not affect the viability of cell at 12.5 and 25 micromolar concentration. However, at 50 micromolar Asiaticoside, the cell viability was reduced significantly. The microscope morphology of the cell remained unchanged. When the cells were treated with 0, 12.5, 25 micromolar. For the cell treated with 50 micromolar Asiaticoside, we observed the shrinkage and scatter morphology of the cells. In our study, the cell could not survive in 50 micromolar Asiaticoside, which contradicts the result of a previous study that used young human periodontal ligament cells. These results may indicate that aging human periodontal ligament cells are more vulnerable than young human periodontal ligament cells. Based on the result of MTT assay, Asiaticoside at 2.5 and 25 micromolar was selected for subsequent experiment. To determine the effectiveness of Asiaticoside on osteogenic differentiation, several osteogenic genes were analyzed using real-time PCR. On day 1, the results show that early gene markers such as bone morphogenic protein 9, LUNGS2, and osteoids were significantly enhanced when the cells were treated with 2.5 micromolar Asiaticoside. On day 7, the early gene markers was not influenced by Asiaticoside. However, the BMP2 level was significantly enhanced at a concentration of 25 micromolar, and BMP1 level was also significantly upregulated at concentration of 2.5 and 25 micromolar. Interestingly, the level of collagen 1 was significantly downregulated. This table summarizes the genes influenced by Asiaticoside. Osteogenic differentiation occurs in 3 stages: cell proliferation, matrix maturation, and mineralization. The middle column shows the genes that were normally expressed during those stages. For the mineralization stage, we use alisalicylate staining to quantify the calcifying nodule when the cells were treated with Asiaticoside for 14 days. The left side presents the accumulation of red calcified nodules under a microscope. The graph on the right-hand side presents the quantification of deposited calcium. At 25 micromolar Asiaticoside, mineralization was significantly promoted. Next, we will analyze our gene expression and mineralization result. We will present only the genes that show alterations. On day 1, the genes that were upregulated were BMP9, LUNGS2, and osteoids. Normally, osteoid expression is regulated by LUNGS2. As a result of increasing level of LUNGS2, the level of osteoid is also promoted. Our result was in contrast with the previous result of Pretty et al., in which the level of LUNGS2 was not altered on day 1, but the level of the osteoid gene was promoted in young human periodontal ligament cells. This may be the result of the difference in the baseline level of LUNGS2 gene, since the LUNGS2 level in aging cells was lower than that in young cells. Since the baseline level of the genes in aging cells is low, it shows a significant difference. Bone morphogenic proteins were in concert with LUNGS2. Our results suggest that upregulation of BMP9 promoted LUNGS2 and osteoids expression. However, the gene expression pattern of BMP2 was maximum on day 7. This may be the cause why BMP9 was promoted on day 1. BMP2 was promoted on day 7 instead. Dental matrix protein 1 was also significantly upregulated on day 7. DMP1 is a non-collagenous transcription protein that functions in the early and late stage of osteogenic differentiation. In the previous study, Asiaticoside was found to promote the expression of DMP1 in human periodontal ligament cells on day 1 and 14. These results suggest that Asiaticoside may influence DMP1 at every stage of osteogenic differentiation. The effect of Asiaticoside on collagen 1 level is interesting. It can either promote or suppress the genes. We hypothesize that it depends on the inflammatory condition of the cells. Keloid fibroblasts and pulmonary fibrosis have high levels of inflammatory cytokines. The aging human periodontal ligament cells also has a high level of inflammatory cytokines compared to young human periodontal ligament cells. This may lead to the suppression of collagen 1 instead of an upregulation.
3 minutes left.
When the upstream BMP2 and DMP1 expression and the upregulation of calcifying nodules at 25 micromolar Asiaticoside. This may suggest that DMP1 and DMP2 play important role in promoting mineralization in aging human periodontal ligament cells treated with Asiaticoside. However, the mechanism of action is unknown. Asiaticoside may activate the BMP SMAD signaling pathway or upstream the number of DMP1 binding calcium ions, leading to phosphorylated DMP1 and transport extracellularly to produce hydroxyapatite. The limitation of our study is that further studies are required to identify the mechanism of action in osteogenic differentiation and mineralization. The effect of Asiaticoside needs to be confirmed in an aging animal model or other in vivo experiments to develop the usage of Asiaticoside in clinical applications. From our results, we can conclude that Asiaticoside can enhance osteogenic gene expression and induce mineralization in aging human periodontal ligament cells. It has the potential to be used as an alternative periodontal regenerative material in clinical application.
1 minute left.
Thank you for your attention.
Thank you for your presentation. You're 1 minute ahead of the time, right? Okay, any question? Would you like to add anything more? Okay, is there any question from the audience? During you are thinking about the question, may I ask a bit? Is there any clinical application for the Asiaticoside in the, have any clinical application before?
They have a study that using Asiaticoside in the form of fiber mat, but using in the normal, using in the wound healing of the skin. So that for now, they still don't have the clinical application of Asiaticoside for treatment in the oral application.
So we have a lot of things, a lot of herbs or plants that we can extract and make use of it to apply in our dentistry, right?
Yes.